Hello, my name is Natalie Rodriguez, and I found that an increase in herbivory caused an increase in foliar secondary metabolites in two types of woody trees, green ash and red pine. So the first graph I'm going to present to you is the flavonoid and phenolic concentrations in the red pine trees. So along the x-axis here we have 1 through 20 red pine trees, and on the y-axis we have the concentration levels. So here flavonoid and phenolics are measured, and the main takeaway is one, that red pine trees are producing secondary metabolites, and two, that the flavonoids are being produced significantly more than the phenolics. So this graph is very similar to the one before, however it is measuring the flavonoid and phenolic concentrations in the green ash tree. So the y-axis here is 1 through 20 trees of green ash, and the y is the concentration levels of the flavonoid and phenolic. So the main takeaway for this one is that 1, the green ash is producing secondary metabolites. However, 2, that we can't really say which one is being produced more in green ash. So you might be asking yourself, why should we care? So the reason why we care so much about secondary metabolites is that they act as a defense mechanism to allow plants to minimize the effects or consequences of an attack. Knowing if plants produce secondary metabolites can allow us to further perform experiments for questions like what type of compounds are being produced and how much is being produced. With these two questions being studied, we could see how secondary metabolites further play a role in plant interactions and plant herbivore interactions. So how I was able to obtain these findings was by using two reports, the first one being called Woody Plant Secondary Chemicals Increase in Response to Abundant Deer and Arrival of Invasive Plants in Suburban Forests. So from the title, you could tell that in this study, we were measuring the secondary metabolites of the green ash trees in response to two herbivores, which were deer, and invasive plant, which was really invasive grass. So the data presented to me, I organized it by dividing it by control and stress data. And then I further simplified the data by only including data that did not include the invasive grass, which is this right here. It says that there was no invasive grass, no invasive grass measured. And the reason why I did not want to include the invasive grass was, one, I just wanted to compare one stressor present instead of two, because the other report only had one stressor, and I didn't want to. I wanted it to be fair. And two, I felt that including two stressors might affect the secondary metabolite concentrations measured, so I did not include them. So this is the flavonoid, and this is the phenolic column, flavonoid, phenolic column concentrations. So this right here is a calculation that I performed. It was a stressor over control calculation, and the reason why I did this calculation was because I wanted to fully focus on the stressor concentration that was measured. So this right here was the flavonoid stressor over control calculation results, and this was the phenolic stressor over control calculation results. And then from there, I ran the ANOVA test because I wanted to see the difference between the two, really focusing on which one was being produced more or not. And from the p-value, we saw that it was a 0.124, which indicated to us that there was no significant difference between the two. Hence why in the beginning I said that we couldn't really tell which one was being produced more. However, we could perform further tests like finding which compounds are being produced and then grouping them between flavonoid and phenolics to further see which one is being produced more. So my second report was called Spatial and Temporal Components of Induced Plant Responses in Context of Herbivore Life History and Impact on Hosts. So in this study, they measured the many secondary metabolites of the red pine in response to the bark beetle that was their herbivore. And again, the way that I organized the data was by dividing it from control and stressed. And this right here is the phenolic concentrations, and this was the flavonoid. And then again, I did the stressed over control calculation, which was right here. ran an ANOVA test and found that my p-value was at a 0.001. So there was a significant difference between the two metabolites, secondary metabolites, flavonoid and phenolics, which tells us that the flavonoid, as shown in the graph before, was being produced more than the phenolic in red pine.